Hi, my name is Raquel Mattos-Canedo. I'm a rising junior at Brown University, and this summer I've had the opportunity to work at the Rojas Moore Lab at The Ohio State University through the NIH SenNet CUSP internship program. And my project focuses on the comparative analysis of cellular senescence in different lung regions. So at the Rojas Moore Lab, we take a look at the lungs specifically, and we work with SenNet to characterize senescent cell variations. So far, studies in the human lung for senescence have exclusively focused on the parenchyma region, which excludes other lung regions that we think are important to look at. So for my project, I decided to focus on three different lung areas: the parenchyma, the peribronchial, and the subpleural regions. So as you can see, each region has a difference in their cell type makeup, as well as differing exposures to the environment and environmental stressors. So the objective of my project is to make a comparative analysis of senescence in these distinct regions, with our ultimate goal being to determine which of the areas is the most susceptible to senescence. So the experimental design for this project works by taking samples from each of the different areas from a healthy young donor. We then inflate those samples with 3% agarose. We incubate it in cold PBS. And then using a vibratome, we slice 400 micrometer thick slices. With these slices on day 0, we change the medium every 1 hour for 4 hours, and then we culture them in DMEM-F12 medium with 1% antimycotic antibiotic and 10% FBS. On day 1, we add bleomycin to induce senescence. Bleomycin is commonly used to induce senescence. It does this through a DNA damage mechanism, and that's what we chose for this project. So then on day 3, we removed the bleomycin and we collected samples on day 7. So for this project, I took samples from 2 donors, age 24 and 29 respectively. And from each of these donors, I had samples from each of the 3 areas. And for each of the 3 areas, I had a control sample and a bleomycin sample. So I made a total of 6 samples per donor for this experiment. So once we had those HPCLS, we pulverized them in liquid nitrogen. It's very important to pulverize in liquid nitrogen before you homogenize to make sure that we are getting a representative sample and to make sure no pieces of HPCLS are sticking to the side of the tubes. So once we had those pulverized, we homogenized in 300 microliters of REPA. We incubated them at 4 Celsius for 30 minutes at 22 RPM. And then we centrifuge them at the same temperature for 30 minutes at 1500 RPM. We then went ahead and quantified these proteins using a 1:2 dilution in REPA. And then we prepared these samples for Western blot using Lemley 10X and REPA. We then ran the gel electrophoresis and we loaded 15 micrograms of samples, which was around 20 microliters. We then ran it at 50 volts for 30 minutes and 120 volts for 1.5 hours. We then used a nitrocellulose membrane for the membrane transfer and used our standard procedure, 25 volts, 1 amp for 30 minutes. And you can see here an example of the Ponceau staining for one of the membranes we ran. So these are the results from the Western blot. We used beta-actin as a loading control to make sure that our data was consistent and we were getting equal amounts of protein in each of the wells. So the Western blot data was really interesting to look at because in the peribronchial region, we saw this significant increase in the expression of both P21 and GDF15, only in the peribronchial region. So that was interesting because in our PCR data, our qPCR data, we also see an increase for the parenchyma region for the senescent markers P16, P21, SERPIN1, and GDF15. So at the transcript level, we're seeing the parenchyma region show significant increase in these markers, whereas with the Western blot, we did not see that at the protein level for the parenchyma region. The peribronchial region, however, did stay very consistent with our protein level analysis with the Western blot. as P21 and GDF15 both showed a significant increase for the peribronchial region. So that was very consistent. Another interesting thing to look at is only the subpleural region showed significant increase in IL-6, which was interesting because the parenchyma region and the peribronchial region did not show that increase for that marker. So from this data, we can conclude that after the senescent induction with bleomycin, the peribronchial area is exhibiting higher protein levels of senescence compared to the parenchyma and the subpleural regions. However, in the transcript level analysis of these areas, we do see distinct patterns in senescent marker upregulation between the three areas. So that was really interesting to reveal. The peribronchial area does demonstrate greater susceptibility to senescence at the protein and transcript levels than the other regions. So for future directions, it would be really nice to run this analysis on old donor samples. This way we can not only compare susceptibility to senescence across three areas, but also compare that to the young donor samples. And from that, we could maybe determine whether one is more susceptible to senescence with age as well. So we would also like to investigate the presence of other senescent markers like P16, P53, and beta-gal, just to further characterize the senescent signature of the three areas in the lung. In closing, I would like to thank the laboratory I was a part of this summer. I would like to thank Dr. Rojas and Dr. Mora for their help throughout this summer, and as well as my postdoc mentor, Dr. Rosas. Everyone in this laboratory was extremely kind to me, and everyone made a very good mentor, and I feel very lucky to have been a part of their work. I would also like to thank the NIH SenNet CUSP internship program. This opportunity meant a lot to me. I walked away from this summer feeling very prepared for my junior year, knowing that I can always use the program as a resource. Everyone was very helpful. I had a wonderful time being a part of this internship. And so I want to say thank you.